Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow at the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the second lecture of the chapter Biomolecules. So till now we have seen about the basic of biomolecules and how can we characterize biomolecules out of a cell. In that we saw different methods, out of which there was one method where we used trichloroacetic acid to solubilize the cell and then to see what fractions come into the filtrate and what fractions come into retentate. Then we also saw how we can characterize the inorganic components of a cell, and that was by using the destructive method. Now, and upon that, what we saw that there are different components such as amino acids, carbohydrates, nucleic acids that were present upon characterization of these components of the cells. Now in this lecture, we will study one of the component that we saw, that is amino acid. Okay. So what is amino acid? Before knowing what is amino acid, let's see what is the source of amino acid. Okay. So how does our body or animals and plants get the amino acids? So you must be knowing that amino acid is the building block of protein, and our body is majorly consist of proteins. But how do our body or any other animal or living organism get this protein? So if we talk about the plants, so in the case of plants, suppose this is a plant. So the plant take inorganic nitrogen. Okay. So it take inorganic nitrogen. In what form? In the form of either nitrate or ammonia. So in this form, plant take nitrogen, and what it does, it synthesizes amino acid using this. So it synthesizes amino acids. Okay. It synthesizes amino acids using inorganic nitrogen, that is in the form of nitrate or ammonia. Now, with this amino acid, the plant cannot, or the animal, any living organism cannot store this amino acid in its original form. So what they do, they convert it into chain of amino acid, that is protein. So the amino acid is converted in the form of protein. Okay. So in this way, a plant synthesizes amino acid by using inorganic nitrogen and then converts it into the protein. But if we talk about the animals, how do human get this amino acids? So for this, what happens? The animals, they take protein in their diet, right? Take proteins from plant in their diet. Okay. And then what they do? Then in the body, we have a digestive system, or animals have a digestive system in which this protein is digested. So the protein that is taken by the plant is digested, and upon digestion, what it forms? It forms amino acids again, because upon digestion, it breaks into its monomeric unit, that is amino acids. This amino acids is now taken up by the cells. The amino acids are taken up by the cells. Okay. And this cell, what the cells do? This cell, it either synthesize new proteins by this amino acid, new proteins. So the cell utilizes these amino acids and synthesize new proteins, or secondly, synthesize new amino acids. So it can synthesize new proteins as well as it can synthesize new amino acids as well. Okay. So this is how an animal and a human gets the amino acids. And then these new amino acids again are converted into or are used to synthesize new proteins. Okay. So this is the source by which the human or the animals get the amino acids. Now, let's talk about the structure of the amino acids. So the amino acids, they consist of they consist of, if you talk about the elements, so it consists of carbon, nitrogen, oxygen, hydrogen, and sulfur. So these are the elements of which amino acids are made up of. Now the second thing, they, as the name suggests, it consists of two terms, amino and acids. So these are the structures that consist of two groups. So they consist of two groups. And what are these two groups? The first one is amino group. Okay. So the first one is amino group. And the amino group is NH2. Okay. NH2 group. And the second is acid, right? So it consists of carboxylic acid group. So it consists of carboxylic acid group, and that is written as COOH, right? So in the structure of amino acids, you will see two groups, the amino group and the carboxylic acid group. Now, the important thing is that amino acids are called as substituted methane. So the amino acids are called as substituted methane. So what does it mean? So we know the structure of methane, right? So methane has 4 hydrogens attached to the carbon. So this is the structure of methane, right? Now in this structure, if you erase the 3 hydrogens, and then in the place of these hydrogens, if you attach one R, this is one functional group, a carboxylic group, and a amino group. So this is known as amino acids. So this is the basic structure. This is the basic structure of amino acid. Okay. In this amino acid, you can see that this NH2 and COH group is present, one hydrogen is present, and there is one R group. Okay. And this R group vary from one amino acid to another. Okay. So if we write about the R group, what we can say that this R group is different. This R group is different in different amino acids. So on the basis of this R group, there are different amino acids. And there are how many amino acids? So the standard amino acids are standard. So the standard number of amino acid is, just a second. So there are 20 standard amino acids. Okay. So standard amino acids I'm showing by AA. So there are 20 in numbers. Okay. Because they have 20 different R groups. Okay. This is R group is a functional group that is different in different amino acids. Now, when you saw the structure of the amino acid here, what you see that here to the carbon, 4 different type of groups are attached, right? So this makes the structure of amino acid asymmetric. So the structure of amino acid is asymmetric. It is asymmetric or chiral. So we know what is chiral carbon. A chiral carbon is the one. Chiral carbon is the one on which 4 different groups are attached. On which 4 different groups are attached. Right? 4 different groups are attached. So this is chiral carbon, or we can say the structure of amino acid is asymmetric. Now, is this true for all the 20 amino acids? Mostly it is true, but there is one exception. Okay. So there is one exception. There is one amino acid which do not have asymmetric structure or do not have chiral carbon, and that is glycine. Okay, so glycine is one amino acid, and if you see the structure of glycine, so what you will find? You will find the basic structure is same. It has COH, it has H, but in this group, in this amino acid, the another functional group is also H. So the R that I was talking about, this is H in the case of glycine. So glycine is do not have a chiral carbon. Okay. Now, Let's talk about one property of a chiral carbon or a chiral molecule. So the chiral molecules are optically active molecules. Okay. You will also study this in your organic chemistry class, that the chiral molecules are known as optically active molecules. And since amino acid is also chiral, so you can write that amino acids are optically active. Amino acids are optically active. Now, what does it mean that amino acids are optically active? It means that they have ability to rotate plane polarized light. Okay, they have ability to rotate plane polarized light. Now, what I mean by this sentence is, you can see through this figure. So the light rays, they exist in different planes. Okay, so they exist in different planes. But when you pass this light through a special material that is called as polarizer, so when you pass the light through the material, this polarizer, what the polarizer do? The polarizer polarizes, or it allows only light of a particular direction to pass through it. So the light that will pass through the polarizer will be in the particular direction. Now, this light is known as plane polarized light. Plane polarized light, because they are in a same plane. Okay, so these all lights are in a same plane. So these are plane polarized light. Now, when this plane polarized light is passed through a solution containing chiral carbon, say a mixture of amino acid, when it is passed through a mixture of amino acids, what you will see? You will see that this mixture will either rotate this light. This mixture will either rotate this light left or right. So the one that is rotated left, is known as levorotatory. It is known as levorotatory, and the one that rotates the light in the right direction, it is known as dextrorotatory. Okay, it is known as dextrorotatory. So this light has the ability, say this mixture of amino acid has the ability to rotate the light either left or right, and hence are known as levorotatory or dextrorotatory. So if we talk about the naturally occurring amino acids, that is all these 20 amino acids. So the naturally occurring amino acids are the naturally occurring amino acids are levorotatory. Okay, so the naturally occurring amino acids are levorotatory, or hence they are. You will also see in the books that it is written as L amino acid. It is written as L amino acid. Okay, so this is the basic or the fundamental about the amino acids. Now the next thing that we'll study is about the zwitter ions. So what are zwitter ions? Okay, so zwitter ions means, the term zwitter ion means that a compound is said to be zwitter ion when it can exist in both positive and negative charge. So if a compound can exist in both positive and negative charge, it is known as zwitter ion. Okay, now when a compound will exist in the positive form or can exist in a negative form, that depends on the pH. Okay, so this nature, whether it is positively charged or negatively charged, nature depends upon the pH of the solution. Okay, it depends upon the pH of the solution in which it is kept. Okay, so based on this, you can say, because the amino acids, they exist as a zwitter ion, so you can say that amino acids have different structure at different pH, right? Because these amino acids can exist as zwitter ion, and because this zwitter ion structure varies with the pH of the solution. So you can say, right, that amino acids have different structure at different pH, right? So the amino acids have different structure at different pH. So what I'm trying to say here is, because this is the structure of your amino acid, you have R group, you have COH, and you have NH2. Okay. Now suppose this is kept in a solution. It is kept in a solution. Now, based on the pH of this solution, say its pH is 4. So based on the pH of the solution, this COH group will either give, or it will take the hydrogen ion from the solution, or this NH2 will donate. Okay, it will donate the hydrogen ion, right? It will either take the hydrogen ion or it will donate the hydrogen ion. And based on that, if it will take the hydrogen ion, it will be negatively charged, right? And if it will donate, it will have a positive charge. So based on that, it can either exist as a positively charged or negatively charged. So if we talk about, say, it's at certain pH, the amino acid is looking like this. Okay, it is negatively charged. Say at certain pH, it is negatively charged. Okay, now because it has the ability to change, and say at another certain pH, it is positively charged. Okay, it is positively charged because it has taken the hydrogen from the solution and it becomes positively charged. Okay, then there will exist a common or a time where this amino acid will have both negative and positive charge, and this structure is known as zwitter ion. So this structure is known as zwitter ion. And at what pH this situation or this structure occurs, that pH is known as isoelectric pH. Okay, so isoelectric pH, what is isoelectric pH? Before that, if you see the structure, what you can see, it has one positive charge, this amino acid, and one negative charge. So if you talk about the net charge on this solution or this structure, what is the net charge? The net charge will be 0, right? Because it has one positive and one negative charge that will cancel each other. So mostly the net charge on the zwitter ion is 0. And the pH at which this occurs is known as isoelectric pH. So what you can write about isoelectric pH? Isoelectric pH is a pH at which net charge on amino acid is 0. Okay, so this is isoelectric pH. Now, this isoelectric pH will also vary from one amino acid to other amino acid, right? So the isoelectric pH vary from one amino acid to another. Okay, so for one amino acid, the isoelectric pH can be 4, while the another amino acid, the isoelectric pH can be 12, right? So it varies from one amino acid to another amino acid, but we understood what is isoelectric pH. It is a pH at which the net charge on the amino acid is 0, or you can say that at this time it exists as a zwitter ion. Okay. Now, the next thing that we'll see is the classifications of amino acid. So there are multiple types of classifications for the amino acid because of the robustness, or you can say because of the structure of the amino acid. Okay. So first type of classification that we have is based on the protein composition. So the first type of classification is based on the protein structure. Okay. So it is based on the protein composition. Okay. So what does it mean? So protein composition means whether the amino acid is the part of protein. Okay. So whatever protein is present in our body, whether that amino acid is the part of the protein or not, based on this, the amino acids are classified in two types. The first amino acid known as standard amino acid, and the second is non-standard amino acids. So what are standard amino acids? These are the amino acids that are part of the protein, part of the proteins. That means that proteins are made up of, or proteins are the chain of these amino acids. Okay. And non-standards are the one, they are not part of proteins. Okay. These are not the part of proteins. Okay. Now, so what are the standard amino acids? So example of standard amino acids are 20 amino acids that we will see. The name of these amino acids we will see in upcoming slides. But for now, there are 20 amino acids that comes under the standard amino acids. And in non-standard amino acids, so in non-standard amino acids, the examples are, there are multiples, out of which you need to remember these two. One is citrulline. And the second is ornithine. Okay. So these amino acids, they do not make the protein. So they will not be present in the chain of protein, but instead they have certain other roles. For example, this citrulline and ornithine, they are used in urea cycle for urea synthesis. Okay. So they are used in urea synthesis. So they are not a part of protein, but they have their own roles. So this is one type of classification that is based on the protein composition, that whether the amino acid is a part of protein or not. Now, the second type of classification, it is based on the R group. So remember the structure in which there is R group, right? And this R group divides the protein, divides the amino acid in 20 types, right? So based on the R groups, the first type of amino acids are aliphatic amino acids. Okay. So these are aliphatic amino acids. So what does it mean? It means that the R group of these amino acids contain chain of carbon, right? They will contain a chain of, you can say contain linear chain of carbon. Okay. So these are aliphatic amino acids. Now, what are aliphatic amino acids? So let's draw one by one. So this is the basic structure of all the amino acid. It is containing an NH2 group, one carboxylic group, and a hydrogen ion. Now, the first example is aliphatic amino acid is alanine. So the R group of alanine consists of CH3. Okay. So this is called as alanine. Okay. Now, which is the second aliphatic amino acid? It is valine. So its R group will have CH3 and CH3. Okay. And this is valine. So the second aliphatic amino acid is valine. Now, the third aliphatic amino acid is CH2CH. Now, just remember the valine in which you will add CH2 at the top. It will become your next amino acid, that is leucine. Okay. So it is leucine. Okay. And the fourth aliphatic amino acid is isomer of this leucine. So we know the isomer, right? They have different structural arrangement, but same chemical formula. So there will be isoleucine, which has the structure of CH3. So it is the isomer of leucine. So its structure has, it has same chemical formula as leucine, but different structural arrangement. And this amino acid is known as isoleucine. Okay. So these are the four. So I was talking about the aliphatic amino acids. So that there are four aliphatic amino acids that are alanine, valine, leucine, and isoleucine. And you can remember this by this all vents look interesting. Apart from this, we also have one aliphatic amino acid in which we don't have chain of carbon, but instead of that, we have hydrogen as the R group. And that amino acid is called as glycine. Right. Now, this is the first type of amino acid category based on the R group. Now the second type is acidic amino acids. Okay, so the second type is acidic amino acids. It means that they have acidic group, or you can say carboxylic group, right? So they have carboxylic group. Okay, so yeah, so they have carboxylic group, right? Now you can say that yeah, all amino acids have carboxylic group, right? But along with the main carboxylic group that is present on the backbone or to the alpha carbon, these acidic amino acids have as additional carboxylic group. Okay, so there are two types of amino acids that are acidic amino acid. The first one is called as aspartate. Okay, so the first one is called as aspartate, and the structure of aspartate is this. So it has CS2 to which a carboxylic group is added. So the R group of aspartate is CS2COOH. Now the second acidic amino acid is called as glutamate. So it is same as aspartate, but it has an additional CS2 group. Okay, so it has an additional CS2 group, and this structure or this amino acid is known as glutamate. Okay, so the aspartate has only one CS2 while the glutamate has two CS2 group. And the acidic amino acids are, so you can write A and G, aspartate and glutamate. These are acidic amino acids. Now the next is basic amino acids. So there are three types of basic amino acids, and this basic amino acids, just like acidic amino acids, they have carboxylic group. The basic amino acids have amino group. So they have additional amino groups. Okay. Now this amino groups can be either 1, 2 or 3. That we will see in the structure. So the first type of basic amino acid is histidine. So what is the structure of histidine? It will have a CS2. Okay. Now see how to remember the structure. You have C, then alternatively you have N, again you have C, then N, then C, and this C is connected by double bond. Okay. Now there you have two double bonds here, so you have alternate C and C and C. Okay, so in this way you can remember the structure of histidine. So the first basic amino acid is histidine. Okay. Now what is the second basic amino acid? That is lysine. So lysine, you have a linear chain of 4 CS2. Okay, you have a linear chain of 4 CS2 and 1 NH3 plus, and this is lysine. Okay, so this is your second basic amino acid, and the third basic amino acid is arginine. So arginine, again it will have CS2 just like lysine. It also has 4 CS2 group, but there is one difference. So you have 3 consecutive CS2, and then you have amino group. Okay, you have amino and C. So you have 4 carbon just like lysine, but the arrangement is different. And what you are seeing the difference is in the case of this arginine. So in the case of this arginine, what you can see, it has 3 extra amino group, right? So because of this reason, it is most basic because it has 3 amino group, and because of this reason, arginine is most basic, while the lysine has only 1 amino group, right? So there are acidic amino acids and basic amino acids. Now the fourth type of amino acid based on R group is amide containing amino acids. So what are amide containing amino acids? They have amide group. So this is what? This is amide group. Okay. Now how to draw the structure? So these amino acids are actually derivative of the acidic amino acids. Okay. So if you remember the structure of acidic amino acid, you can easily remember the structure of these amide containing amino acids. So the first is, it is derivative of aspartate. So, and because of this reason, it is known as asparagine. Okay. So the name is asparagine. Now, How will you draw the structure of asparagine? First, draw the structure of aspartate, that is CS2CO. Okay, CO. Now, instead of OH, you have amide group attached to it. Okay, so instead of OH, you have amide group attached to it, and it will become asparagine, right? Similarly, if you want to, the second amide containing amino acid is derived from glutamate, and its name is glutamine. Okay, so its structure is also simple, right? First, you draw the structure of glutamate, that is CS2, CS2, CO, and instead of OH, now you add NH2. So this is your glutamine. Okay, so what we saw? We saw that there are, till now we have seen that there are 4 types of amino acids based on the R group. Now the next type is sulfur containing amino acids. So as the name suggests, that these amino acids have sulfur in their R group, right? And there are basically 3 types of this amino acids, out of which 2 are standard. Okay, we saw what is standard and non-standard amino acids. So there are 3 sulfur containing amino acids, but out of 3, 2 comes under standard amino acids. And what comes under standard amino acid? The first is known as cysteine. So the structure of cysteine is something like this. Okay, so this is cysteine. Okay, so it has CS2 and now the second sulfur containing amino acid is methionine. Okay, second sulfur containing amino acid is methionine. So its structure is CS2, CS2, S and SH. Okay, so this is methionine. This is methionine. Now these 2 are standard amino acids. Okay, cysteine and methionine. Now the third amino acid is cysteine. The name is also cysteine, but this is non-standard. So the next I am going to draw is non-standard amino acid. So non-standard amino acid. And what does the structure has? It has, you can write the non-standard amino acid and structure has 2 cysteine. It has 2 cysteine connected by disulfide bond. Okay, it has 2 cysteine connected by disulfide bond. So how does this structure look like? So you have 1 cysteine. Now you connect it with a disulfide bond. Okay, so this is sulfur of 1 cysteine. This is sulfur of another cysteine. It is connected to CS2 and it is connected to backbone, right? It is connected to, one second. Yeah, it is connected to backbone, right? So this is the structure of cysteine. Okay, so there is difference in the spelling of both this cysteine and this cysteine. So this cysteine is what? It is 2 cysteine connected by a disulfide bond, and this is the disulfide. This is the disulfide bond. Okay, so these are sulfur containing amino acids. Now next what we will see hydroxyl containing amino acids. So what are hydroxyl containing amino acids? So for sulfur containing, you can remember it like CCM, right? Cysteine, and methionine. Now again, like sulfur containing amino acids, we have 3 hydroxyl containing amino acids. Okay, so the first hydroxyl containing amino acids is like cysteine only. So if you remember the structure of cysteine, it has CS2SH. Similarly, if we talk about the first hydroxyl containing amino acid, you draw the structure CS2, but instead of SH, you write add hydroxyl group, that is YH. And then this amino acid will become serine. Okay, this is the R group of serine. So it is same as cysteine, but it has OH instead of SH. Now the second is threonine. So the threonine also contain this hydroxyl group, and its structure looks something like this. Okay, so instead of having OH in the end, it has OH in the beginning, and this structure is known as threonine. Yes, so this structure is known as This structure is known as threonine. Okay, so it is known as threonine. So this is threonine. Now the next hydroxyl containing amino acid is proline. Okay, sorry, tyrosine. So the next is tyrosine. So what does a tyrosine look like? So this tyrosine has one unique feature, that it also has aromatic ring. So it also has aromatic ring. So the tyrosine, it comes under both category, the one that is hydroxyl containing amino acid as well as in aromatic ring containing amino acid. So it also comes under aromatic ring containing amino acid. Okay, because it has aromatic ring. So this is the sixth type of the amino acids based on the R group. Now the last type is cyclic amino acids. So what is cyclic amino acid? It means that the R groups of these amino acids contain cyclic ring, right? Cyclic ring. What is cyclic ring? So you must have studied or will study in the organic chemistry class that it must have certain cycles, something like this or something like this, right? So these kind of structures must be present there in their R group. Now in this, let's see what are different types. So the first is phenylalanine. So phenylalanine, if you see the name, what you can see, it has phenyl and alanine. So we know the structure of alanine, right? Alanine has CH3 here, right? Now if you want to draw phenylalanine, what you'll have to do? You'll have to remove one hydrogen, so it will become CH2, and add phenyl group here. And this is phenyl group, right? This benzene ring. So it will become phenylalanine. Now the second is tyrosine. So we just saw the structure of tyrosine. So tyrosine, what we have, draw the structure of phenylalanine, and because tyrosine is hydroxyl containing amino acid, add OH here. So it will become tyrosine. So now we have phenylalanine, tyrosine. The next amino acid is tryptophan. So the structure of tryptophan is quite complicated, and also it is one of the complex amino acid. So let's see how does tryptophan looks like. Also, its structure is not very important, but for your knowledge, the tryptophan looks something like this. Okay, it will have a group like this, and NH. So this structure you have to practice. So a tryptophan has a structure like this. Okay, and then it has benzene ring attached to it. Okay, it has benzene ring attached to it. So this is the structure of tryptophan. This is tryptophan, and tryptophan is what? It is most complex. It is most complex. Okay. Now there are 5 cyclic amino acids, out of which we have drawn 3. Now the next is histidine. So histidine we structure also saw earlier, right? So histidine is also positively charged or basic amino acid. And structure is what? You have CS2, then C. Then you have alternate C, N, C, N, then C, right? So this is the structure of histidine. Okay, this is the structure of histidine. So histidine, we know it is positively charged, or you can say basic amino acid. The basic amino acid or positively charged amino acid, right? And it also have it also comes under cyclic amino acids. Apart from this, we have one more cyclic amino acid that is called as proline. But the structure of proline is quite different as compared to all the 19 other amino acids. How it is different, you will see. So it also have a chain. Okay, it also have a chain. But what is different in this? You remember this amino group that is common in all the amino acids, and the amino group is free, right? But in the case of proline, this amino group, it forms a bond with the R group or the side chain. Okay, so the structure looks something like this. So this is proline, and it is the only amino acid in which the amino group, the amino group, an amino group forms bond with R group, right? So the amino group that is present in the backbone, it is forming a bond with this R group, right? So this is the only amino acid. Okay. So we saw there are five cyclic amino acids. Now, if you see at the structure carefully, you can see some of these amino acids contain aromatic rings. So we know what is aromatic ring, right? That contain a benzene chain, right? So the benzene chain looks something like this. Okay. So they are the amino acid that contain this aromatic ring. So this is aromatic ring, right? This is aromatic ring. So which of these are the amino acids that contain aromatic rings, or they can also be categorized as aromatic amino acids? We can see through the structure, right? The phenylalanine, it has this benzene ring, right? So the first is phenylalanine, right? Now, which is the second amino acid that contain benzene ring or aromatic ring? Tyrosine, right? So we have tyrosine that contains aromatic amino acid. And the third, if you see tryptophan, the tryptophan also consists of aromatic ring. This ring is aromatic ring, right? So the third is tryptophan. So these are aromatic amino acids. Okay. Now there is one more concept, that is heterocyclic amino acids. So what is heterocyclic? Heterocyclic means it consists of two types of atoms in their cycle, okay, such as carbon and nitrogen that we can see. So from the figure, what we can see, that we have two heterocyclic amino acids. The first one is tryptophan. Why? Because tryptophan, if you see in their cycle or in their ring, it has carbon as well as nitrogen. So it is heterocyclic. Different types of elements is present in the ring. So the first one is tryptophan, and the second you can say is histidine, because the histidine also have a two N in their ring. Okay, so the heterocyclic amino acids example are tryptophan and histidine, while the aromatic amino acids are phenylalanine, tyrosine, and tryptophan. So with this, we completed the classification of amino acid based on R group. So based on R group, what we saw that there are 7 types of or 7 categories of amino acids. Now, the next classification is based on polarity. Okay. So what do we mean by polarity? Polarity means that the amino acid exist or have charges on their either side. So because of that, it creates a pole, right? Or because of this, these amino acids have hydrogen donors or acceptor group. Okay. Or acceptor group. Because suppose it has a negative charge, what it will do? It will take the hydrogen from the solution, right? So it will become a hydrogen acceptor. Or suppose it has a positive charge, then it will remove the hydrogen from its group, and hence it will become hydrogen donor. And based on that, there is a polarity of that amino acid. Now, before categorizing the amino acids based on the polarity, let's see what actually polarity means, or what the role of the polarity. So if you see the structure of amino acid, what we see that we have this thing, right? This group, COH, and then we have R group. So this is an amino acid in isolation, or a single amino acid. But like I said in the first slide of today's lecture, that amino acid do not exist as in their original form, but they are converted into proteins, right? So how these amino acids are arranged in the protein? So if you see, so in protein, the amino acids exist something like this. So they will exist something like this. Okay. COCNH. Okay. So this is, and here you have NS2. You have R group, R group, R group, right? NS2. Okay. So here what you are seeing, this is one amino acid. This is your one amino acid. Similarly, this is your another amino acid. Okay. Now these amino acids are connected to one another via this bond, and this bond is known as peptide bond. Right. So what you are seeing here, the peptide bond is being formed between the carboxylic group of one amino acid and amino group of another amino acid. So this carboxylic group and amino group, they are involved in peptide bond formation, right? So what is free in this case? So in the protein, what is free? We have this R group. This R group is free, right? And this R group decides the polarity of that amino acid. Okay, we understood why. Because this carboxylic group and amino group are involved in the peptide bond formation, and only thing that is free is this R group. And because of this R group decides the polarity of amino acids. So we can write that R group, the R group decides the polarity of amino acids, right? Because this R group is free, and because of this reason it will form bond with the solution in which it is present, and hence it decides the polarity of amino acids, right? So if you divide the amino acid based on the polarity, you will divide it by keeping in the mind the R group that is present on that amino acid, not this NH2 and CO group that is present on the alpha carbon. So one more thing, this carbon on which this R group is attached is known as alpha carbon. Okay, so we decide the polarity based on the R group that is present on the amino acid. Now, if we talk about the classification based on the R group, there are two types of classification. The first one is non-polar, or second one is polar. So the non-polar, they do not have polarity. Okay, and to remember this, you can remember by LAMP, VIP, GT. Okay, so these are the amino acids that are non-polar. So let's write the name. So the L stands for leucine, right? A stands for alanine. M stands for methionine, methionine. P stands for proline, and V stands for valine. I stands for isoleucine. P for phenylalanine. P stands for phenylalanine, alanine, and lastly, G stands for glycine, and T stands for tryptophan. So these are non-polar amino acids. Okay. Now, if we talk about the polar amino acids, now in the polar, we have different subcategories. In the polar, the first one is charged. The first one is charged amino acid. So what are charged amino acid? Again, the charged amino acid can be either acidic, and the examples we saw, that was glutamate and aspartate, right? That was glutamate and aspartate. And the basic amino acids is also charged. So acidic means negatively charged, right? Acidic or negatively charged. And basic, or these are positively charged. Okay. So in the basic, what we had histidine, we had lysine, and arginine, right? So these are charged amino acids. Now the second category is in the polar is uncharged. So these are polar but uncharged. They do not have charge. Okay. So these are examples are serine, hydroxyl group containing amino acid, that is serine, and threonine. Then we have sulfur containing cysteine. Then we have one more hydroxyl containing, that is tyrosine, because they have hydroxyl group. Okay, so the solution will be polar, the amino acid will be polar, but they do not have any charge. Okay. Then we have amides, that is glutamine, and asparagine. Okay, so these are the examples of polar amino acids. and these are non-polar amino acids. Okay. So based on polarity, the amino acids are divided into two types, polar and non-polar. For non-polar, you can remember by LAM, BIP, and the remaining will be polar. Okay. So this is another type of classification based on polarity. Now, the last classification of amino acid, that is based on nutritional requirements. Okay. So based on nutritional requirements, the amino acids are divided into two types. What do I mean by nutritional requirement? So whether they are required in the diet or not. Whether required in diet or not. So based on this, we have three categories of amino acids. The first one is essential, the second is semi-essential, and third is non-essential. So essential, as the name suggests, so they are required in the diet, right? Required in the diet. So essentials are the ones that are essentially required in the diet. Now, semi-essentials, not very important, but required in special cases. So these are required in special cases. Okay, we'll see what are the special cases. So these are required in the special cases. And non-essentials means they are not required in the diet. So if they are not present in the diet, it's fine. Okay, so not required in the diet. Okay, these are non-essentials. Okay, now why they are required? Why the essential amino acids are required in the diet? Because our body or cell cannot make these amino acids. Okay, cell cannot make these amino acids. So because our cell cannot make these amino acids, hence they are required in the diet so that they can be taken from outside. The semi-essentials, the cell can make it. Okay, cell can make these, but they do this very slowly, but at very slow rate. Okay, so they can be made by the cells, but they are made at a very slow rate. And non-essentials, cells can make these amino acids. Okay, so the cells can make these amino acids, hence they are not required in the diet. Now, what is the example of essential amino acid? We'll just see. But for now, just write the example of semi-essential amino acid. So the semi-essential amino acids are histidine, and arginine. Okay, so these amino acids are semi-essential because our body can make these amino acids, but at a very slow rate. And hence, in certain cases, for example, in case like pregnancy, so in pregnant women, these amino acids are given as a supplement from outside. If we talk about athletes, right? So athletes, because they have to do very heavy exercises, so these amino acids are made at a very slow rate, they are supplemented from the outside. Okay, so in these cases, this amino acid becomes semi-essential, or they are required to be given from outside. Okay, now one more thing. In case of plants, we saw in the first slide, right, that a plant is able to make all the amino acid by itself. So in the case of plant, all amino acids are non-essential. This is case of plant. So this is the case of in plants. So in plants, all amino acids are non-essential because a plant is able to make all the amino acid by itself. If we talk about the essential amino acids, there are a total of 9 amino acids. Okay, there are total 9 amino acids that are essential, out of which in animals only 7 are essential. 7 are essential. Okay, 7 are essential. Now, if you look at the essential amino acids, what we can see, we can remember it by this: I love my little hairy tiger very passionately and truly. So now let's write what it means. So I stands for isoleucine. Okay, So it is essential. So these are essential amino acids. Essential amino acids. Okay. So isoleucine. Next is leucine, methionine. We have methionine. We have lysine. We have histidine. Now, this histidine, in certain cases, you will see it will be under essential amino acid, but it depends upon the situation. Okay. Now you have tyrosine. Sorry, here you have tryptophan. Tryptophan. You have valine, phenylalanine, and what we left with threonine. Okay, so these are essential amino acids: isoleucine, leucine, methionine, lysine, histidine, tryptophan, valine, phenylalanine, and threonine. Out of this histidine, and in some cases you will also see that histidine and arginine people have written in essential amino acids, but these are semi-essential. So you can, based on the question, you can omit this. Okay. Now I was talking in the case of animals, what are 7 essential amino acids? So you can remove out from this. You can remove for animals. What you can do? You can remove head. Remove head. So what is head? Head is histidine, alanine, and threonine. Okay. So if you remove these 3 amino acids, then the rest 7 are essential amino acids in the case of animals. Okay. So with this, we have completed the classification of the amino acids. Now in the next lecture, we will see about the roles of amino acids, and we will also see certain questions. Then we will move to the proteins. That's all for now. Thank you.